Brilliant. Awesome. Thank you very much. And thanks everybody for being here. It's my first time here as well. And so far, really, really cool. So I'm at NASA Ames, just down the road, literally. And I'm in the exobiology branch. Exobiology was what astrobiology used to be called back in the day. But bottom line is we're interested in searching for life on other planets. And today's lecture is about a place that is very special. Maybe not everybody here has heard of it, but I think that it's probably at the forefront of exobiology and the search for evidence of life in the solar system. And I'm hoping I'll convince you by the end of the lecture that this is probably the best place to go if you're searching for evidence of life, alien life. So everybody should know this place. This is Mars. When I started working at NASA almost 20 years ago, this was the only place for exobiology, really. This was a place that we knew had been probably habitable in the past. It was very Earth-like. There were evidence of river channels on the surface, probably even oceans way back then. And so everybody was focused on the possibility of life on Mars. But in the last 20 years or so, there's been a little bit of a revolution in our understanding of other places in the solar system. And in particular, there is this family of what we now call ocean worlds. And raise your hand if you've heard about ocean worlds at some point. Awesome, great. These are fascinating bodies. These are icy moons. Most of them are orbiting the giant planets. You will probably recognize Europa. There is a mission going there. Europa Clipper should get there in about 5 or 6 years. This is Europa, sorry. There is Callisto and Ganymede. We've known these places as icy moons and ocean worlds for a while. The first indications that Europa was an ocean world go back to the Galileo missions in the 1990s. There is Titan. Everybody knows Titan. What a fascinating place. There is Enceladus, my favorite place. I'm going to talk about it today. And then there is Sirius, which is actually not a moon. It's a dwarf planet in the asteroid belt. It's not technically an ocean world. It's a candidate one. We don't fully know if it's an ocean world, but it's fascinating because there is evidence on the surface, and you can see there, of salts that were excavated from the inside. And when you see salts, that equals water. It means that at some point, or even today, Sirius has a lot of water in the inside. So really interesting places. And each one of them is different. They are all icy moons. They're frozen on the surface, but the interiors are probably very different, each one of them. And each one is an opportunity to understand chemical processes that are in a very different environment than what we're used to, even on the surface of Mars. And those chemical processes could be actually very relevant to the surface, to the possibility of life elsewhere, as I'll try to show you. So this is Enceladus. It's a little moon. It's about 250 kilometers in diameter. You can fit it between San Francisco and LA. That's how small it is. This is a picture of Enceladus just floating over the rings of Saturn, and it was taken by the Cassini mission. Cassini was a mission that launched in 1997, made it to Saturn in around 2004 or so, and its objective was to study Titan and Saturn and the rings of Saturn. But shortly after Cassini arrived to the Saturn system, they flew by Enceladus and they discovered something truly unexpected and fascinating. They discovered that the south pole of Enceladus has these massive cracks. They're called tiger stripes. They're giant fractures in the south polar terrain. This is another image taken by Cassini. And by the way, this is an image of the Saturn family where you can see Enceladus. And for those of you who are connoisseurs of astronomy, there is one of the rings of Saturn is called the E ring, which is the faint ring of Saturn. Turns out, as you will see in a second, the E ring is actually due to things happening on Enceladus. And that's very cool. But what Cassini discovered was actually a plume of material coming out from the inside of Enceladus. Enceladus is emptying itself outside into space. And you can see images of the plume, that it's actually in the form of jets coming out. So in 2005, Cassini discovered this plume. It actually triggered a reimagination of the entire mission so that it could actually fly through the plume and collect samples of those materials to analyze them chemically with the instruments that the spacecraft had on board, and you'll see results from those measurements. Cassini flew through the plume 22 times, collecting samples and analyzing those samples. The plume, as I said, is made of jets coming from the surface, Each one of those jets has been mapped to the tiger stripes. It's about 100 of them. And then there's also steady emission of gas. It's called curtains. So you have the jets on the one side, you have the curtains on the other side. So Cassini had a bunch of instruments, but two of them were particularly interesting for today's talk. Both of them could carry chemical analysis of any materials that came inside the instruments. There's three things that I want to highlight today of things that Cassini detected. The first one are gases and simple organic compounds. You'll see these in a little bit. The second one are complex organic compounds. When you talk to an exobiologist, organics is like, that's how you get us interested. This is the stuff of life. And then hydrothermal emissions. And I'll tell you in a second why we think that there is hydrothermal activity and what that means for the possibility of life there. So let's walk through each one of them. These are the gases. There's not too much, I mean, it's really interesting. I don't want to spend too much time talking about this, just to point a couple of things. One is that most of the gas in the plume is made of water, 96 to 99%, but there's also hydrogen, CO2, methane, and ammonia, and probably other things that the instruments were not sensitive enough to detect. Now, H2 and CO2 are very interesting because this is food for some microbes here on Earth. Microbes on Earth, some of them can feed on H2 and CO2. And if you've noticed something about Enceladus, is that it doesn't have an atmosphere. And it's too cold, so there's no liquid water on the surface. But there's water coming out because we've seen the ice from the inside. And H2, and what that means is that the inside of Enceladus is dark. If there's any life in there, cannot use sunlight as an energy source, which is what most of the biomass on our planet comes from, is from sunlight. But the fact that there is what we call a redox pair, a pair of chemicals that can react and life can make them react and extract energy from it, fulfills one of the requirements for life. That's an energy source. That's why we got so excited about H2 and CO2. And then there are little organics that were detected early on in the mission, little organic compounds. Now, what's really interesting is in the solid phase. So the plume, as I said, is made of jets and curtains. The curtains are made of gas. The jets are made of ice, little icy grains, about a few microns in size or even smaller. And Cassini could analyze those icy grains that are ejected into space. There are three basic types of grains. They call them originally type 1s, type 2s, and type 3s. Type 1s, and that's based on their chemical composition. Type 1 grains are mostly made of water, almost pure water. So really not very interesting. We won't spend too much time talking about them. Type 2s, this is where the good stuff is. Organic compounds, so water plus organic compounds. Some of those organic compounds are relatively small, like the ones we saw in the previous slide coming with the gases as well. But some of them are really, really big. And when I mean big is the types of organics that we expect to find in places like these are in the scale of mass for molecules. We call them Daltons. They go from one to many. We typically look for organics that are 50, 60, 70, 80 Daltons. That's when you start to see organics that are relevant for biochemistry, building blocks of life, and whatnot. We see some of those organics, little ones, but then we see things that are 3,000 Daltons in size, 4,000 Daltons in size. You need a lot of chemistry to make those organics happen. You don't need life. That can happen in the absence of life. But the fact that we see complex organics means that there is something really fascinating going on in that little moon that we don't fully understand because we don't have a lot of information about those organics. But the organics are there, and they're being ejected into space in these type 2 grains. And then finally is the type 3s. And the type 3s are the ones that are telling us where is the water coming from? Where are those icy grains coming from? The type 3s contain salts. And as I told you a second ago, where you see salts, that means liquid water, because salts is actually what water leaches out of rock. And if you have salts, that means that rock is in contact with liquid water, and that's where your salts are coming from. So when you put all this together, you have this plume of gases and icy grains, mostly made of water with organic compounds and salts, which point to something happening in the inside of the moon that involves liquid water. And then Cassini also found these little silica grains. Silica is an element, almost pure silica nanograins, also coming out in the plume. And what silica is interesting why it's interesting is because silica typically forms in hydrothermal vents. When you go to the bottom of the oceans on Earth, there are hydrothermal vents. This is a picture of a hydrothermal vent with the black smokers and whatnot. And if you collect samples of this stuff coming out from the vent, among other things, you can find these little silica grains. So finding silica grains on the plume is an indicator that it might be hydrothermal activity inside of the moon. Okay, so really cool stuff. Now, you put everything together, and this is our model of what's going on Enceladus. You have an icy crust, which is about 5 to 10 kilometers. It changes from the equator to the poles. That crust is broken up in fractures, and through those fractures, there is stuff coming out, the jets and the curtains. What's feeding the plume? It's a large ocean of liquid water, about 60 kilometers deep. At the bottom of that ocean, hydrothermal vents, very similar to the hydrothermal vents that we find here on Earth. And then you have the rocky core, about 200 kilometers worth of it, actually 400 kilometers worth of it. So this is literally an ocean world. It's mostly actually made of rock, but it has a lot of liquid water in it. There is, I think, by people's estimates, as much water in Enceladus as there is water in the oceans of the Earth. And so for an exobiologist, for somebody who's interested in life elsewhere, water, energy, and what we'll see in organics are the ingredients for life. Doesn't mean there is life there. It means it's a really interesting place to look for it. So when putting everything together, this is just what I said, hydrothermal activity. We have the stuff coming out in plume. We have the icy grains with oceans, with organics embedded in them. And then we have all the conditions required for habitability conditions. The elements for life, the energy, H2 and CO2, an environment that is compatible with life. The temperature of the water is about 0 degrees C. The pH is estimated about 8, which is not bad for life, 8 or 10. The salinity is slightly lower than the salinity of the oceans of the Earth. So it's a place where most of us actually would be happy campers. So the question then is, fascinating place in its own right. The question then is, could there be life there? And what I do is I come up with strategies to search for life. So when NASA says we want to search for life there, they come to not just me, but others as well, and they ask, what do we measure? What do we look for? What would be an indicator of life? They actually ask, if we have to spend $3 billion and we want it to succeed, what really do we want to measure? Because failure is really not an option. So typically, if you ask an astrobiologist what to search for when we search for evidence of life, they'll tell you biosignatures. Biosignatures are things that only life can make. There's some examples here. There could be imprints on a rock of life that existed a long time ago, or there could be minerals or biominerals that microbes can generate, and they have certain sizes and distributions that are only life can actually make. Or could be stromatolites, which you've probably seen on TV in documentaries, which are fabrics of microorganisms that existed a long time ago, and now they're turned into rock, but we can see evidence of them in their structure and in their composition. It could be certain molecules, like oxygen in the atmosphere. There's a lot of oxygen in our atmosphere, and that's indicative of biological activity. Or complex molecules that only life can make, or mixtures of gases, or even swimming cells. All of these things are biosignatures. And if you ask 10 astrobiologists which biosignatures we should be looking for in places like Enceladus, you'll probably get 15 different answers, because it's really complicated. It's not as simple. There are problems with biosignatures, and we need to be aware of them. Number one is science risk. That's what I call science risk. When you formulate a mission to search for life, and what you're looking for is something that only life can make, then you're defining your mission success as a binary proposition. You go there and you find it, you succeeded. You go there, you don't find it, you failed. And there you go, $4 billion. And a Congress hearing, probably. If you're familiar with the history of planetary research, there was a mission going to Mars in the 1970s called Viking. Viking was the first lander on Mars, successful lander on Mars. There were some crashers, but this was a lander. And Viking had several biological experiments in it, which successfully collected soils of dirt from the surface of Mars, incubated it in those experiments, looking for evidence of biology. And to this day, there is debate in the scientific literature whether Viking discovered life on Mars or not. Most people think that the experiments failed. A few people still think that maybe you could explain it biologically, but that's an example of that's an example of things that we don't want to happen. We don't want to spend 50 years debating whether what we looked for was biological or not. And this is part of the science risk of looking for biosignatures. The other one is what I call mediocrity bias. If you're searching for something that only life can make, you're implicitly assuming that life actually took place, it existed. The origin of life took place in this environment. And we don't really know how life originates in the first place. We don't even know how life originated on Earth. We don't know how likely it is. So Enceladus could be perfectly habitable, but maybe it never had the conditions for life to evolve in the first place. So again, that would be another reason why your search for biosignatures could fail, just because life never originated. Another one is confirmation bias. You're looking for things that you want to find, and that's not good science. It's like looking for your keys under the light. As a scientist, you try to develop hypotheses that they're not preferential towards a specific outcome. And where you're searching for something that only life can make, you're assuming that life is there and I'm going to find it. And that's bad practice. And then you open up yourself to false negatives. You think that life maybe makes crystals or compounds or looks like this, and maybe it doesn't, because we don't really know what life elsewhere might look like. So maybe you're looking for something that we terrestrial organisms make, but organisms on Enceladus, if they exist, maybe they don't make that compound or that structure, and you would conclude that there is no life there, when in fact there is life. It is just measuring the wrong thing. That's a false negative, and we don't want false negatives. So for all these reasons, I think we need to think differently about how we search for life. And it's gonna get a bit technical if it's not technical enough for you so far, but bear with me for a second. This is a really simplified story of how life originated on Earth. If anybody ever asks you, how did life originate on Earth? Well, it started with very simple compounds. And if you pay attention, some of them we've mentioned already: hydrogen, CO2, water, nitrogen compounds. Those, in an environment where it's chemical disequilibrium, which means that there might be rocks in the environment, metals, things that can catalyze chemical reactions, these simple compounds can react and form little small organic compounds. Remember, small organic compounds in the plume of Enceladus. Those small organic compounds, further reaction, they can turn into things that we start to be familiar with because these are things that life makes as well. Amino acids, sugars, nucleic bases, which are the things that our DNA and RNA are made of, carboxylic acids, which are the precursors of cell membranes. All this chemistry can happen spontaneously. All this chemistry has been found, for example, in meteorites. There's really nothing mysterious about it. What's mysterious is what happens afterwards. Somehow, these compounds, somehow, somewhere on Earth, they started joining together and making what's called polymers, longer chains of those compounds. Things like peptides, the precursors of proteins. Things like nucleotides, the precursors of RNA and DNA. Phospholipids, the membranes, the precursors of the membranes. And then through more and more chemical evolution, these things become more complicated, and at some point they get encapsulated, and now you have something that is a protocell, something that it's almost life. And at some point there is a transition, which is still mysterious, and this thing starts to evolve by itself. Darwinian evolution, natural selection, all that good stuff. And then it diversifies, and it leads to all the beautiful diversity of life that we see here on Earth. So this is, in a nutshell, our best guess at how life originated on Earth, and how it might originate elsewhere. And these are the steps that this process takes. It's a lot more complicated than this, but this gives you a good idea. Now, on Enceladus, we have chemical disequilibrium. We've proven that. And we have complex organics, which could form from this process. They could form from biology. We don't really know. So these two things, these two check marks, we can say, yeah, we found this on Enceladus. We haven't found any evidence of any of the other steps. Now, the question of searching for life becomes then, where along this process are you going to start searching for? What are you going to search for? Most biosignatures, they sit up here. Once life takes hold and diversify, then it starts to make all kinds of crazy things. Some biosignatures, they sit here. DNA, RNA, complex proteins, complex peptides, they sit here. I advocate for looking even farther down, earlier on, this type of chemistry. And I'll give you a few reasons why that's probably the best place in this whole chemical space to search for evidence of life. Nothing that you see here means life until you start looking at specific details, and then you can start to tease things out. Now, what's funny and interesting to me is that this chemistry, except for this little part at the bottom, all this chemistry, none of this chemistry has ever been demonstrated in a habitable environment outside of the Earth. And that is a huge knowledge gap, because what this means is that all this is supposition. It's all model-based. It's all imagination. Not until we go somewhere else outside of the Earth and we start to tease apart this chemistry and find evidence for the different pieces of this puzzle, then we can be sure that this is a sound model for how life originates. So I say we might as well start at the beginning, because that's probably the easiest place to start. This is also an area that is interesting because that's an overlap between prebiotic chemistry and biochemistry. So you, me, elephants and trees and bacteria, we all make these types of compounds. But they can also be made spontaneously in the absence of life. We call them abiotically or prebiotically. And what this means is that if we go to Enceladus, two things can happen. Either there is life there or there is no life there. If there is life there, we expect life to make at least some of these things just like we do. If there is no life there, we expect other processes, abiotic ones, to make some of these things. So there is really low science risk. We will find something. We're not hedging our bets only on finding something really extraordinary. So we will get some data. There will be understanding coming out of it. The question is whether we're smart enough to tease out whether these things that we find, whether they're mean biology or not. So the science risk is less. The fact that these things are also very abundant and they're easy to form means that when life forms anywhere in the universe, it's very likely that it's going to start with these same building blocks. And so it's going to use, if not all of these, at least some of these compounds. So there is likely to be an overlap in our biochemistry and the biochemistry of forms of life on Enceladus if they exist. And that overlap is likely to be here. It's less likely to be up here, because what happens here is evolution, and that is planet-specific. There is no reason why life on Enceladus would be giving science lectures on a Friday. This is an Earth invention, and we cannot assume that happens elsewhere in the universe, because it takes a lot of evolution for that thing to happen. But we can be certain, or relatively certain, that there might be a biochemical overlap down here, because this is stuff that is very everywhere in the universe. It's very easy to make. All right. Hang on. Okay. So this is one of the favorite things that this is what, if I had $5 billion, this is what I would have spent it on. Searching for amino acids on Enceladus. What a proposition. Amino acids are little compounds. That's what we use to make proteins of. You might be familiar with proteins. You might not be familiar with amino acids. Amino acids are the building blocks of proteins. And these are some examples of amino acids that we use to build proteins. The simplest one is glycine, and then there's alanine, and there are more complex ones with side chains and things like this. But all of them have the same structure. There is two carbons, and then there is a side group that can be complex or can be simple, and then there is a nitrogen group somewhere there, and then there is an acid group on this end. So there is the general structure. We use all life on Earth, every organism uses 20 amino acids to build proteins, and it's always the same 20. We all share the same alphabet of amino acids, and it's 20 of them. And when you join two amino acids, they actually that's what happens. That's what needs to happen to get a protein. They join in what's called the peptide bond, and then you have two amino acids linked, and then there comes another one, another peptide bond, and then you keep expanding this chain until you make a protein. And then you can do all kinds of interesting chemistry. Now, amino acids in nature most amino acids come in two flavors. We call them L and D, right-handed and left-handed. They have exactly the same structure, but they're mirror images of each other. So it's essentially the same compound chemically, but their structure has mirror images. It's fascinating that life on Earth, all life on Earth, only uses the L form to make proteins. Nobody knows why. Somebody made a choice 4 billion years ago. I like L, and now we're all stuck with L. But that is important because it gives us a way to identify whether an amino acid is biological or not, or a mixture of amino acids. Because when you make amino acids in the absence of life, spontaneously through abiotic reactions, like the ones we find in meteorites, we find 50-50 abundances, because the universe doesn't care. It's just as easy to make D as it is to make L. So they tend to form at roughly equal abundances. But life, for some reason, they like L, and that's all we make. And so if you go to a place and you search for amino acids, and you find them, and you measure their chirality, that's the property, the L versus D, and all of them happen to be one or the other, that's very hard to explain in the absence of life. That could be an indicator of life. I'm giving you a hint of the kinds of things that I would like to measure in places like Enceladus. Now, another fascinating thing that I'm fascinated by this fact, these are the 20 amino acids that we use to build proteins. I'm not going to name them, but these are the 20. And these are the average frequency in which these amino acids appear in life on Earth. There is like 7,000 organisms pulled into this plot. And as you can see, some of them are in slightly higher abundance, but roughly they are pretty much in the same abundance. The amino acids that you see in red, these are, I call them prebiotic, because these are what we find commonly in meteorites, in asteroids, in comets, Urey-Miller experiments, if you remember from high school, electric spark experiments, those were produced as well. If you want to make amino acids, these are the ones that you're going to make with. The ones in blue are biosynthetic only. These are inventions of Earth biology. These things don't happen in the absence of Earth biology. We came up with those. So roughly half of the amino acids we use, they are an endowment from the prebiotic world where life on Earth originated. Those were lying around, and the first protocells, the first cells, used them because they were available, and that's how the first proteins were mostly based on these prebiotic ones. And then through evolution, we started adding new amino acids because they helped us solve problems that life encountered through evolution on Earth. This is important, as I mentioned earlier, is because if you're going to search for life elsewhere, you want to search for things that are likely to be there at the time of the origin of life. So you want to focus on the ones that are in red, the ones that are prebiotic. If you're searching for amino acids that only life can make, then chances are that life on Enceladus, if it exists, actually came up with a different set of amino acids. There are hundreds of possibilities out there, and there's no reason why the same alphabet should exist on Enceladus, same alphabet of amino acids. There is reasons to believe that at least some of the prebiotic ones would be chosen by those forms of life. So now we're narrowing down the types of things that we're looking for. We're looking for amino acids, yes. We're not looking for all of them, we're looking for some, and then we're looking for properties of those amino acids that are influenced by the activity of organisms. One property is what I call the abundance distributions. So in this plot, you see the relative abundance, the concentration, the abundance of amino acids, some of those that I mentioned, in a natural sample, as a function of the size of the amino acid, what's called the carbon number. The higher the carbon number, the bigger the amino acid. And as you can see, in this specific plot, there is a lot of 2-carbon amino acid, the simplest one, glycine. The next one is slightly less, and then less, and then less, and by the time you get to 6 carbons, you don't find any. That's an exponential decline in concentration. And that's what you expect to see in an abiotic environment. The universe is lazy, as I told you. It doesn't care about L's and D's. I'll make the same stuff, and I'll make a lot of the little ones, and I'm not going to make a lot of the big ones, because it's easier. There's less energy required to make these than to make that. So the abiotic chemistry tends to make a lot of the little ones. On the other hand, this is the same plot, but out of organisms, out of life on Earth. And as you can see, the distribution is much different. This is because we need to invest energy in solving problems. If I need to run that hill, and to run that hill, I need to make a big amino acid, I'm going to make the amino acid, otherwise the lion is going to kill me. It's not quite as simple, but that gives you an idea of how evolution works. We're willing to invest energy in creating new things that didn't exist in nature, even if they're big ones, just because they serve an evolutionary purpose, a survival purpose. Okay? So we can search for amino acids, we can focus on the ones that we expect they're going to be there, and then we can measure their abundances, and then we can see whether their distributions look like something like this, or their distributions look like something that is not this. If they look like this, I can explain that abiotically. I can say, well, I know how to make that in the lab. If it looks like this or something that is very different, then I can say, this is really exciting. We've never seen that before in the absence of life. Let's look further. This is a case study. We actually took the effort to go through the literature and look for all the publications where they had measured amino acids in meteorites, in simulated Titan organic material. That's samples made in the lab. Ryugu is an asteroid, so those are samples that actually we returned from an asteroid and analyzed in the lab, and they contain amino acids. And hydrothermal simulations. These are chemical simulations in the lab that simulate hydrothermal vents. In all these cases, the little amino acids, glycine and alanine, are the simplest ones. When you pull them together, they make almost 75% or more of the total amino acid composition of the sample. All other amino acids, low abundances. This is kind of what I showed you before. A lot of little ones, few of the big ones. A lot of little ones, few of the big ones. What happens when you look to many places on the planet and you do the same analysis? You look at organisms, you look at soils and permafrost and all kinds of samples from different places. You look at aquatic environments, you look at actual hydrothermal vents at the bottom of the ocean. You can see a more even distribution, just because life is actually spending energy building things that it needs to survive. So one might say this is a biosignature. Yeah, it's a signature of biological processes. I don't like to call it a biosignature. It's a source diagnostic property that if we find this, it means one thing. If we find that, it means another thing. Either way, we go to Enceladus and we look for them, we should find one or the other. We just need to cross our fingers and hope there is the one that we want to see. Now, you can make the same exercise for other compounds or even for amino acids. Remember, I told you amino acids come in L and D forms, so you could look for which one is represented, if both are represented equally or not. You can look for the abundances. I just mentioned that. Now you have two independent ways of testing whether there is something going on there that is shady, sketchy. We cannot explain it abiotically. You can look for other compounds, like lipids, the things that we make our membranes from. And the same logic applies. The distributions of lipids, when they form abiotically, follows one trend. When they're formed biologically, they follow a different trend. And there is more information embedded in it. So these are the types of things that when they ask me what to look for when we search for life, these are the types of things that we recommend. Now we need to build the instruments to do that, and we need to build the missions that are going to be able to obtain those samples. And I'll just close the lecture with mentioning one mission concept that we came up with a few years ago. We named it Orbilander, originally another original name, because it's a spacecraft that would go to Enceladus. It would originally orbit around Enceladus, and then the same spacecraft would land on the surface. So it's an orbiter and a lander, all in one, Orbilander. And this is the spacecraft there, and it has a big funnel, because it's like we're just going to sit there and stick our tongues out and wait for all that ice to fall back on the surface, just as you do when it snows. And then we're going to ingest that sample and run it through the instruments inside, and among other things, look for amino acids and look for lipids and measure their handedness and measure their abundances and see what those distributions tell us about processes happening in the ocean. Now, this is a summary of what that mission would do. It would not just do that. It would actually do 5 different independent life detection measurements, looking at organics, the bulk organic composition, look at the amino acids, look at the lipids, and then it would look for 2 of those biosignatures, just because we had some astrobiologists in the team that they wouldn't let go of their favorite biosignatures. So, okay, we'll put their biosignatures in there. A polyelectrolyte is a molecule like DNA or RNA, so something very complicated that only life can make. And then cell-like morphologies, microscope, see if we can, some of those samples we have swimming things in there. Unlikely, but very cool if we would find it. And then there were a whole bunch of other instruments that would look at the structure of the moon, the interior of the moon, like seismology, ground-penetrating radar, things like this, so we could actually see the ocean with data, and we could see the rocky core, and maybe we could even see ocean currents if they're taking place, and even this ice shell and the piping where all these plume materials are coming from. So it will be a very, very cool mission. Yeah, we have some time. Just for those in the room that are interested in this, especially the younger ones, it takes about 12 years to get to Enceladus the moment you launch. Stop smoking, start exercising, eat your veggies because it's a long mission. This one, in this iteration, it would launch in 2038. It would make it to Enceladus in 2050. It would start its science phase around 2052. Funny enough, throughout the entire 2040s, the south pole of Enceladus is going to be in the winter. It's a 10-year winter, just because of the way the orbits work. And it's going to be in the shade. It's going to be in the shadow. So really, no point landing there if you can't see where you're landing. So we can't land any sooner than that. Either we launch tomorrow, or we need to wait until 2038, and then the sooner we can land is sometime in the early 2050s when the sun starts to come out again in the south pole. So that's one of the reasons. But it still takes about 9 to 10 years to get to Enceladus. It's a long trip. And then you spend about 3 years slowing down before you can go into orbit around Enceladus, because it's such a small moon. You really need to go slow for Enceladus to capture you in orbit. By the time we make it to Saturn, we're flying about 20 miles per second, and we need to slow it down to a couple hundred meters per second. And to slow down, we spend 3 years in the Saturn system, flying by all the moons, not all the moons, but many of the moons. Every time we fly by one moon, that moon pulls the spacecraft a little bit through gravity, and that slows it down a little bit. Then we know another flyby, another pull, and it sheds off some more velocity. And after dozens and dozens of flybys and 4 years, now we're flying slow enough that Enceladus can capture us, and then we can go into orbit. Then we'd spend a couple of years in orbit, sampling the plume, doing all kinds of science, and then we land on the surface. It actually does a backflip, if you can believe it. NASA engineers, phenomenal. It does a backflip, and then it lands on the surface. And then it spends 2 years, 1 year, 2 years on the surface, doing more of the edges of the science with the funnel, also with a shovel, collecting soil, ice and plume materials, ingesting them, and doing all the measurements that it needs to do. Price tag, $2.5 billion. That's 2025 dollars. So it's an expensive mission, but that's a very cool mission too. Now, a few years ago, there was a meeting of the National Academies where a whole bunch of scientists got together to produce what's called the Decadal Survey. That's 10 years worth of recommendations to NASA of what to do. This is what we think NASA should do the next 10 years in terms of science and missions. And Orbilander was ranked as the second highest priority flagship mission. Flagship missions are the most expensive missions that NASA builds. James Webb Telescope was a flagship. Mars Sample Return would be a flagship. These types of missions. If you're asking, the top priority was the Uranus Orbiter and Probe. Cool mission, not a search for life mission. We were second. So does it mean it's going to fly? We don't know. Things are changing, as you probably can imagine. There's going to be another decadal between now and 2038. That decadal might come up with other recommendations, or maybe at that point the community says, enough is enough, let's go somewhere and search for life because it's way too cool. So I think this is where I'm going to stop. Thank you very much. We have 20 minutes and change, or even less, for questions, and we already have one, so yeah. Yeah.
Was going out to this moon because it has this particular feature, right, with water. But it's a very expensive mission.
Yeah.
You mentioned Mars.
Yeah.
Why can't we do some of that work in Mars?
Okay.
Like the work you were referencing. What can't we do it there?
Yeah. So the question is, why can't we do this on Mars, which is faster? I'm not going to say cheaper, but certainly we've done Mars exploration before. A couple of things. Mars today, at least on the surface, anywhere we can actually access on the surface of Mars, and that is the surface and maybe a couple of meters depth, it's uninhabitable. There are no reasons to believe that life could exist there today, extant life. It's too cold, it's too dry, the radiation is too high, liquid water is not stable on the surface. So you don't have any of those conditions that I mentioned. The science community has pretty much known that for a while, since Viking. Viking tried, didn't find anything. So the effort on Mars has been on going to places that were habitable 4 billion years ago. Places that we know there were lakes in the past, or because we see the sediments and we see the chemical composition, and look for fossil evidence of life, things that would be, remnants of life. imprints on a rock, or certain organics that survive for 4 billion years, or what have you. That would be certainly complicated. It's very hard. Mars sample return was supposed to help us on that. So you bring the samples back from a place that could have hosted life way back then, and then you throw the kitchen sink of instruments that you have here on Earth looking for the little traces of life. That, to me, as an exobiologist, don't quote me, or I'll get fired, I don't think it's good enough. I don't think it's not scientifically satisfying. Because I'm not searching for life for the sake of searching for life. I'm searching for life on other planets because I want to understand the fundamental laws of biology. We have an N of one, an example of one form of life on Earth. It's all life on Earth. We're all the same biochemically. We're all made of DNA, 20 amino acids and RNA, the same chemistry. And so it's really hard to answer fundamental questions about biology with an N of one. I want to know, is every form of life depend on DNA? Do you always need DNA and proteins? Is there always a cell membrane? Do we always do the same types of chemical reactions? Does everybody evolve through natural selection? All these fundamental questions we will never be able to answer until we find life 2.0, another form of life. And a fossil is not going to give you the information. The information is in the actual meat, in the microbe, in the living microbe. We need to find it, kill it, dissect it, and analyze it. So a dead microbe that is fossilized is not good for us. We need to kill it fresh. It's like your fish. And then you can start to tease it out and understand what makes it tick. And then we can start to tease out those fundamental laws of biology. So that's why I would rather go there than Mars. Yeah.
Why do we make the assumption that all life will be hydrocarbon-based?
Yes, carbon-based. Yeah, carbon and hydrogen-based. So it is an assumption. I think it's a solid one. Carbon-based life is compatible with water, or rather, water is compatible with carbon-based life. A different type of chemistry, people talk about silicon-based life, not quite so good in water. It doesn't have the same chemical flexibility. Carbon happens to be a compound that is stable enough that I can be here giving a talk without decomposing or combusting spontaneously. But it's also unstable enough that can change over time. I used to be this tall and now I'm this tall. And I can break down carbon compounds and eat them, and then I can make more carbon compounds with them. Silicon is a lot more stable. Look at a rock, it's made of silicon, doesn't change a lot from one day to another. And so that's another thing. To make these compounds like proteins that carry out chemical reactions, it's very hard. Carbon is very good at these carbon strings of carbon molecules, because at some point in water they will bend, they will twist, and they will form these three-dimensional shapes. That's what actually carries the chemistry of proteins. Silicon ones, they don't bend quite as easily. And really there is no other alternative that people can think of. So, plus carbon is one of the most abundant elements. After hydrogen, helium, and a few others comes carbon, hydrogen, oxygen. So I don't think I think it makes sense that life on Earth is carbon-based, and I think it makes sense that probably any form of life is going to be carbon-based. Finally, I know how to detect carbon-based life. I don't know how to find silicon-based life. So I will start with the places that I know, and when we run out of places that are water-based and carbon-based, maybe we can start thinking about more exotic things. But we really need to think about how to search for those forms of life, but I wouldn't tell you how. So, yeah.
Which other places have good probability of finding life, just like
Enceladus is at the forefront because we have all this wealth of data collected by Cassini that we know. So we can build a mission with some data and be purposeful about it. Most likely Europa is the next one. We don't have quite as much data. Europa Clipper will visit. Europe is on the way there. It has instruments similar to Cassini. It might be able to also find little plumes if they exist, look for organics. If that is the case, then Europa and Enceladus will be head-to-head. Maybe Europa with a slightly advantage, maybe, I don't know, because it's bigger and maybe it's more stable than Enceladus. Enceladus is small, so maybe weird things happen in its history. People talk about Titan and talk about weird life. Titan is an ocean world, it's an icy moon, just like Enceladus, but it's covered by like a few kilometers worth of organic matter and some lakes of hydrocarbons in them. People astrobiologists are inventive enough that some can come up with ideas of methane-based life and things like this. I don't think it's feasible, but there is a subsurface ocean of liquid water in the interior of Titan, and that could be another interesting place if we knew how to access it. Problem is access. And then at that point, then you have your Ganymedes and your Callistos and all these other moons. We wouldn't run out of places. I expect one. I would be happy if every generation gets to visit one moon and search for life. So our generation, I say Enceladus, the next one Europa, and the next one, I'll be dead, so yeah, you guys hash it out. Yeah.
So we see the plumes and we get all this data. Has there been any talk, like, if there were similar plumes coming from Earth, are we surprised with what we found or didn't find?
So are we surprised with what we found in the plumes?
Or didn't find, if you imagine that Earth would shoot up similar plumes, would we expected to see certain signature?
The instruments that were on Cassini, they were not made for plume sampling, they were made for other things. And they also had, it's 1990s technology, so they were limited in their sensitivity and their resolution. So they could only take like coarse pictures, chemical pictures, so to speak, of the chemistry. It's not enough to tell whether it's expected or unexpected. It's like the elephant in the dark room, and it's like you're just touching one part of it. And is it an elephant or something else? It's just enough it's enough of a tease to make everybody very excited. But it could be anything. We don't really know those really big macromolecules that the Cassini found. We have an idea of what they're made of. It's not entirely unheard of. It's hydrocarbons. We find it in meteorites. So in that sense, it's not really weird. But the discovery of the plume itself, with chemistry in it, and the fact that we could turn around a mission and redesign it so it could actually analyze it, that was the spectacular thing coming out of it. It's certainly the closest we've ever been to something that could be complex organic chemistry of the types of things that we find on Earth. So yeah, that's just, yeah.
You said that all life on Earth have the left-handed amino acids.
For protein building, yeah.
Protein building, so and it was just kind of a fluke, or it just happened one way or the other.
Yeah, we don't know why.
So
Yeah.
if it was the other way.
Mirror life, that would be awesome. If we go to Enceladus and they're all D, I would ask them, why D? Does it taste better? I don't know.
But does it also set you in a different trajectory or something?
Not necessarily. So we can actually make D proteins on Earth in the lab. You can make proteins just like the ones we use, but with the D form, which are used, for example, for drug delivery, because your body will not recognize them. They not fight it. And so you can easily carry those drugs with D proteins. If I put an artificial L protein in your body, your antibodies are going to latch onto it and attack it. But if it's D, it's like, oh, we don't care, it's D. Or maybe it's like, oh, D. And so it's you can have mirror life. There is no reason, biochemically speaking, that we can think of that D life wouldn't work. In fact, our DNA is made of D sugars. So proteins, L amino acids, DNA, D sugars. Something was going on at the origin of life, like, come on, make up your mind. Pick one. But no, it was D sugars and L amino acids. The opposite would work just the same.
Why don't we do one more question and then move on to the next part of our...
Oh man, there are two. There's three. Okay, well.
Let's go with that side.
Okay, we haven't done this side. All right, let's go.
What would be the dimensions of this spacecraft that would go up?
Orbilander, oof. Sorry, I turned it off. Orbilander was 6 tons wet mass, that means with fuel, 2,000 dry mass with 140 kilograms. So this would probably be, the antenna I think is about, no, the cone is 1 meter across. This cone is about 1 meter across. This antenna is about 3 meters across. So 3 meters, 6 meters. I don't know, you work in feet, I work in meters, but, and then a few more meters, so 20 feet across or something like this.
Then how many people could be in there?
No people. No. I mean, if you volunteer, it will be a tiny place. Imagine 12 years inside that. It will be pretty horrible. But no people. It will be its own thing. And it's not coming back. So, I mean, you can still volunteer. To each his own, but yeah.
Okay, well, why don't we thank Dr. Davila?
All right. Thank you.